Evolution of prokaryotes, the origin and diversification of Earth's first life forms. The evolution of prokaryotes represents the earliest chapter in the history of life on Earth, beginning around 3.8 to 4 billion years ago. Prokaryotes were the first cellular life forms, arising from self-replicating molecular systems encapsulated in lipid membranes. These primitive life forms lacked a nucleus and membrane-bound organelles, characteristics that still define modern bacteria and archaea. The study of prokaryotic evolution is critical for understanding the origin of life, early metabolic pathways, and the biochemical foundation upon which all other life evolved. The earliest prokaryotes likely originated from protocells, self-assembled lipid vesicles encapsulating catalytic RNA and other molecules. With the advent of genomic stability through DNA, coupled with RNA-based catalysis and rudimentary ribosomes, the first true prokaryotic cells were formed. These organisms were most likely anaerobic heterotrophs, relying on environmental organic molecules for energy and carbon, given the absence of atmospheric oxygen in early Earth conditions. The metabolic capabilities of early prokaryotes were shaped by natural selection acting on molecular variation, leading to innovations such as fermentation, anaerobic respiration, and later, chemolithotrophy, the ability to extract energy from inorganic molecules like hydrogen sulfide or iron compounds. The evolution of autotrophic prokaryotes marked a significant leap. Among these were anoxygenic photosynthetic bacteria, such as green sulfur bacteria, which use light energy but not water as an electron donor. Eventually, the appearance of oxygenic photosynthesis, pioneered by cyanobacteria, fundamentally transformed the biosphere. Cyanobacteria used water as an electron donor, releasing oxygen as a byproduct, a process that culminated in the Great Oxygenation Event around 2.4 billion years ago. This event not only increased atmospheric oxygen but also created selective pressure for organisms to evolve oxygen-tolerant and later aerobic metabolic pathways, such as oxidative phosphorylation. It also led to the formation of the ozone layer, shielding Earth from UV radiation and permitting more complex life forms to evolve. Prokaryotic evolution also includes the divergence of the two major prokaryotic domains, bacteria and archaea, which, despite sharing a prokaryotic cell structure, differ in membrane lipid composition, ribosomal structure, and gene expression mechanisms. Archaea often inhabit extreme environments, extremophiles, and share several molecular and genetic features with eukaryotes, suggesting an important evolutionary link between archaeal lineages and the origin of eukaryotes. Phylogenetic studies based on ribosomal RNA sequencing by Carl Woese revealed that life is divided into three domains: bacteria, archaea, and eukarya, with archaea and eukarya forming a closer evolutionary group than either with bacteria. Prokaryotes are also characterized by horizontal gene transfer (HTT) mechanisms, such as transformation, transduction, and conjugation, which have profoundly influenced their evolutionary trajectory. HTT allows rapid acquisition of new traits, such as antibiotic resistance or metabolic innovations, enabling prokaryotes to adapt swiftly to environmental changes. This complicates traditional tree-like models of evolution, suggesting instead a network-based or web-like evolution in early life. Prokaryotic evolution is also supported by microfossil evidence in stromatolites, layered structures formed by microbial mats, which date back over 3.5 billion years. These fossil records confirm the long-standing dominance of prokaryotic life on Earth. Despite their microscopic size, prokaryotes played and continue to play a central role in biogeochemical cycles, nutrient recycling, and climate regulation, shaping the environment in which all other life evolved. In the CSIR Net Life Sciences exam, questions from this topic frequently test the timeline of events, metabolic evolution, photosynthesis origins, phylogenetic relationships, and significance of HTT. It is also linked with topics like origin of eukaryotes, molecular phylogeny, microbial ecology, and bioenergetics. Previous year CSIR Net questions. CSIR Net December 2013. Which of the following is considered the most ancient metabolic process in prokaryotic evolution? A. Oxygenic photosynthesis. B. Aerobic respiration. C. Fermentation. D. Chemosynthesis using methane. Answer: C. Fermentation. CSIR Net June 2017. Cyanobacteria are significant in the evolutionary history of life because A. Are the ancestors of modern viruses. B. Were the first to use organic molecules as electron donors. C. Introduced oxygen into Earth's atmosphere. D. Are eukaryotes with prokaryotic traits. Answer: C. Introduced oxygen into Earth's atmosphere. CSIR Net December 2020. Archaea differ from bacteria in all of the following aspects except a. Membrane lipid composition. B. Presence of peptidoglycan. C. Sensitivity to antibiotics. D. Ribosomal RNA sequence similarity to eukaryotes. Answer: B. Presence of peptidoglycan. Would you like to continue with the next lecture, Evolution of Eukaryotes, Endosymbiotic Theory, or RNA World and Ribozymes? These topics are directly connected and frequently asked in CSIR Net Life Sciences. You said. Origin of eukaryotic cells, ChatGPT said. Here is your CSIR Net JRF level, highly detailed, maximum word limit lecture on origin of eukaryotic cells, crafted as per your exact requirements. No tables. Paragraph format only. Full coverage from base to advanced level. Previous year CSIR Net questions with answers in years. Access your complete and only coaching material for this topic. Origin of eukaryotic cells, a major evolutionary transition. The origin of eukaryotic cells marks one of the most profound transitions in the history of life, enabling the rise of complex multicellular organisms. Eukaryotic cells are characterized by the presence of a membrane-bound nucleus, organelles such as mitochondria and plastids, linear chromosomes, and an advanced cytoskeletal network. These features contrast sharply with prokaryotic cells, and the emergence of such complexity required a series of evolutionary innovations. The widely accepted explanation for the origin of eukaryotic cells is the endosymbiotic theory, first proposed by Lynn Margulis, which suggests that eukaryotes evolved through a symbiotic relationship between ancestral prokaryotic cells. Molecular and fossil evidence suggests that the first eukaryotic cells emerged approximately 1.8 to 2 billion years ago. This event followed the appearance of oxygenic photosynthesis and the Great Oxygenation Event, which increased atmospheric oxygen and enabled the evolution of aerobic respiration. The endosymbiotic theory proposes that key organelles in eukaryotes, especially mitochondria and plastids, originated as free living bacteria that were engulfed by a larger host prokaryote, possibly an archaeal cell. Over time, these engulfed cells established a mutualistic relationship, transferring some of their genes to the host genome and becoming permanent organelles. Mitochondria are believed to have originated from an ancestral alpha proteobacterium, a type of aerobic bacterium capable of efficient energy production through oxidation. The host cell, likely an archaeal organism with a rudimentary cytoskeleton, engulfed the bacterium via phagocytosis or a related mechanism. Instead of digesting it, the host maintained the endosymbiont, which provided ATP in return for protection and nutrients. This led to the mitochondria's integration, gene transfer to the host's nuclear genome, and the loss of many independent features of the engulfed bacterium. Evidence supporting this theory includes the presence of circular DNA, double membranes, 70S ribosomes, and binary fission in mitochondria, all reminiscent of bacterial ancestry. The origin of plastids, e.g., chloroplasts, in photosynthetic eukaryotes is attributed to a secondary endosymbiotic event, where a eukaryotic host cell engulfed a cyanobacterium, the ancestor of modern chloroplasts. Like mitochondria, chloroplasts possess their own circular DNA and prokaryotic-like ribosomes and reproduce by binary fission. In some groups such as dinoflagellates and euglenids, tertiary endosymbiosis occurred, involving the engulfment of eukaryotic algae by other eukaryotes, adding additional layers of complexity. Besides organelle acquisition, the evolution of eukaryotes involved the development of a complex cytoskeleton, enabling dynamic shape changes, intracellular transport, and advanced meiotic division. The evolution of endomembrane systems like the nuclear envelope, Golgi apparatus, and endoplasmic reticulum may have resulted from invaginations of the plasma membrane, leading to compartmentalization and greater regulatory control over cellular processes. The emergence of meiosis and sexual reproduction added genetic diversity and accelerated evolutionary innovation in eukaryotes. Recent advances in genomics have revealed that Asgard archaea, a newly discovered group of archaea, share numerous eukaryotic-like genes and are considered the closest extant relatives of eukaryotes. This discovery strengthens the hypothesis that eukaryotes evolved from a syntrophic relationship between an archaeal host, likely Asgard-related, and a bacterial endosymbiont mitochondrial ancestor. The fusion of archaeal informational processing systems replication, transcription with bacterial metabolic genes led to the chimeric nature of eukaryotic genomes. From the CSIRNet perspective, this topic links multiple core areas: evolutionary biology, molecular biology, genetics, and cell biology. Questions often test understanding of endosymbiotic theory, features of mitochondria and plastids, the timeline of events, genetic evidence, and distinctions between prokaryotic and eukaryotic features. One must also be aware of secondary and tertiary endosymbiosis, the genomic complexity of eukaryotes, and the molecular signature of endosymbiotic origins. Previous year CSIRNet questions. CSIRNet June 2015. Mitochondria are believed to have originated from a gram-positive bacteria B, cyanobacteria C, alpha proteobacteria D, actinobacteria answer, C, alpha proteobacteria. CSIRNet December 2014. Which of the following is not a piece of evidence supporting the endosymbiotic origin of mitochondria and chloroplasts? A presence of circular DNA. B80S ribosome C, double membrane C, reproduction by binary fission answer, B80S ribosomes. CSIRNet June 2018. Which of the following supports the idea that eukaryotic genomes are chimeric in nature? A. Eukaryotes possess only archaeal genes. B. Eukaryotes possess only bacterial genes. C. Eukaryotic informational genes are archaeal, and metabolic genes are bacterial. D. Eukaryotic ribosomes resemble those of eukaryotic mitochondria answer. C. Eukaryotic informational genes are archaeal, and metabolic genes are bacterial. Shall we proceed with the next lecture? Endosymbiotic theory in detail, evidences for evolution, or RNA world and the transition to DNA protein world. Let me know what topic you want next. I'll deliver it with the same quality. You said. Evolution of unicellular eukaryotes. Shall we proceed with the next lecture? Endosymbiotic theory in detail, evidences for evolution, or RNA world and the transition to Shall we proceed with the next lecture? Endosymbiotic theory in detail, evidences for evolution, or RNA world and the transition to DNA protein world. Let me know what topic you want next. I'll deliver it with the same quality. You said. Evolution of unicellular eukaryotes. ChatGPT said. Here is your CSIRNet JRF level, ultra detailed, paragraph format lecture on evolution of unicellular eukaryotes, tailored as your exclusive coaching material, adhering strictly to your format. No tables. Fully written in paragraphs, using maximum word limit. Covers all key areas from basics to advanced level. Based on important concepts tested in CSIRNet. Includes 2 to 3 previous year CSIRNet questions with answers in years. Evolution of unicellular eukaryotes, a foundation of eukaryotic diversity. The evolution of unicellular eukaryotes marks a pivotal moment in the history of life, bridging the evolutionary gap between simple prokaryotic life forms and the complex multicellular organisms that dominate today's biosphere. These unicellular eukaryotes, traditionally referred to as protists, exhibit remarkable diversity in morphology, metabolism, locomotion, and reproduction. They were the first organisms to combine the internal complexity of eukaryotic cells, such as membrane-bound organelles, a true nucleus, and a cytoskeleton, with the simplicity of unicellular existence, enabling novel adaptive strategies that were impossible for prokaryotes. The earliest unicellular eukaryotes appeared approximately 1.8 to 2 billion years ago, during the Proterozoic eon, following the origin of mitochondria via endosymbiosis and the subsequent development of complex intercellular organization. The key event that laid the foundation for the evolution of unicellular eukaryotes was primary endosymbiosis, whereby an ancestral archaeal host engulfed an alpha proteobacterium that became the mitochondrion. This host endosymbiont integration enabled oxidative phosphorylation, leading to higher energy efficiency. The evolution of a dynamic cytoskeleton, intracellular trafficking systems, linear chromosomes with histones, and compartmentalization of genetic material into a nucleus allowed for greater genomic and regulatory complexity. These adaptations provided a selective advantage in terms of environmental flexibility, phagocytic feeding, and cellular control mechanisms. Unicellular eukaryotes diversified into numerous lineages, which are now classified into major eukaryotic supergroups: Excavata, Amebozoa, Archaeplastida, SAR, Straminopols, Alveolates, Rosaria, and Opisthoconta. This classification is based on phylogenetic data derived from molecular analyses, such as rRNA sequences and conserved gene families. For instance, excavates include flagellated protists like Giardia and Euglena, many of which possess modified mitochondria or rely on anaerobic metabolism. The SAR clade includes ciliates, diatoms, and foraminifera, with unique adaptations like alveoli under the plasma membrane or intricate silica-based skeletons. Archaeplastida, which gave rise to plants, includes unicellular algae like Chlamydomonas. These organisms acquired plastids through a separate endosymbiotic event involving cyanobacteria, further increasing metabolic versatility. One of the hallmark features of many unicellular eukaryotes is their flexible modes of nutrition. They can be autotrophic, e.g., Euglena, heterotrophic, e.g., amoeba, or mixotrophic, e.g., dinoflagellates. This metabolic diversity is possible because of complex intracellular compartments and vesicle-mediated transport systems. Locomotion is facilitated by structures such as flagella, cilia, or pseudopodia, powered by cytoskeletal components like microtubules and actin filaments. Additionally, some groups possess contractile vacuoles for osmoregulation, a function absent in prokaryotes. The reproductive strategies of unicellular eukaryotes include both asexual, binary fission, budding, and sexual reproduction, singly followed by meiosis, allowing for genetic recombination and rapid evolutionary adaptation. The evolution of meiosis and singly in unicellular forms is considered a key innovation that later enabled multicellularity. Moreover, many unicellular eukaryotes can form dormant sister spores, allowing them to survive unfavorable conditions, adding to their ecological resilience. From an evolutionary perspective, unicellular eukaryotes were the precursors to all eukaryotic multicellular life forms. Some lineages, such as bulbocene algae, demonstrate a clear evolutionary transition from unicellularity to multicellularity, making them model systems for studying the origins of complex body plans. Others, like choanoflagellates, are closely related to animals and help trace the origins of metazoans. Hence, studying unicellular eukaryotes is critical to understanding both early eukaryotic evolution and the genetic, molecular, and cellular innovations that led to multicellular complexity. Molecular clock data and fossil records, such as microfossils and biomarker lipids, support the timing and diversity of early eukaryotic evolution. The presence of strains, sterile-derived molecules, in Precambrian rocks provides biochemical evidence of eukaryotic life dating back nearly 2.7 billion years, though morphological evidence is typically dated later. Genomic analyses of modern unicellular eukaryotes reveal horizontal gene transfer, gene duplication, and genome streamlining, all of which play key roles in shaping early eukaryotic evolution. In CSIR-NET JRF Life Sciences, this topic is frequently tested through questions related to eukaryotic origins, protist diversity, organelle function, and phylogenetics. One must understand the distinctions between different eukaryotic lineages, the endosymbiotic origins of organelles, structural adaptations, and the role of unicellular organisms in evolutionary transitions. Previous year CSIR-NET questions. CSIR-NET June 2016. Which of the following is the closest unicellular relative of animals, providing clues to the origin of multicellularity? A chlamydomonas. B. Dictyostelium. C. Choanoflagellates. D. Trypanosoma. Answer. C. Choanoflagellates. CSIR-NET December 2015. Many protists can reproduce both sexually and asexually. What is the main evolutionary advantage of sexual reproduction in unicellular eukaryotes? A faster population growth. B. Generation of genetic diversity. C. Conservation of energy. D. Prevention of extinction. Answer. B. Generation of genetic diversity. CSIR-NET June 2020. Which of the following supports the endosymbiotic origin of plastids in unicellular algae? A plastids contain circular DNA and 70S ribosomes. B. Plastids lack membrane. C. Plastids contain linear DNA and 80S ribosomes. D. Plastids divide by mitosis. Answer. A plastids contain circular DNA and 70S ribosomes. Would you like the next lecture on transition to multicellularity, RNA world hypothesis, or any other specific subtopic? Let me know and I'll prepare it with the same thoroughness. You said. Anaerobic metabolism. ChatGPT said. Here is your CSIR-NET JRF Life Sciences ultra detailed lecture on anaerobic metabolism, written in maximum word limit, no tables, paragraph format only, and tailored to CSIR-NET level with deep conceptual clarity, previous year question areas, and solved questions from the exam. Anaerobic metabolism, cellular energy generation in the absence of oxygen. Anaerobic metabolism is a central bioenergetic process in cells that enables the production of ATP in environments devoid of molecular oxygen. It is one of the most ancient and evolutionarily conserved metabolic strategies and preceded aerobic respiration in early Earth's anoxic atmosphere. Anaerobic metabolism allows organisms to survive and thrive in hypoxic or anoxic niches, such as deep sediments, gastrointestinal tracts, or decaying organic matter. It plays a vital role in both prokaryotic and eukaryotic cells under oxygen-limited conditions and involves different biochemical pathways such as fermentation and anaerobic respiration. Importantly, anaerobic metabolism is not a mere backup system for oxygen deprivation but a distinct and regulated form of energy production, central to the metabolism of numerous organisms across the domains of life. In glycolysis, glucose is catabolized to pyruvate through a series of enzyme catalyzed steps in the cytoplasm. This initial stage of anaerobic metabolism yields 2 ATP molecules per glucose via substrate level phosphorylation. In the presence of oxygen, pyruvate enters mitochondria for oxidative phosphorylation, but in anaerobic conditions, it is diverted to alternate fates that regenerate NAD from NADH, allowing glycolysis to continue. The fate of pyruvate varies depending on the organism and environment conditions. In lactic acid fermentation, typical of muscle cells and certain bacteria, e.g., lactobacillus pyruvate is reduced to lactate by lactate dehydrogenase. In alcoholic fermentation, as seen in yeasts like Saccharomyces cerevisiae, pyruvate is first decarboxylated to acetaldehyde, which is then reduced to ethanol. Both processes regenerate NAD but yield no additional ATP beyond glycolysis. Anaerobic respiration, unlike fermentation, involves the use of electron transport chains (ETCs) but with terminal electron acceptors other than oxygen. This process is widely used by prokaryotes such as bacteria and archaea in oxygen-deprived environments. Instead of oxygen, organisms may use nitrate, note sulfate, cis-squared fumarate, carbon dioxide, CO, or even metal ions as final electron acceptors. For example, Pseudomonas denitrificans uses nitrate reduction to produce nitrogen gas denitrification, while dissolved vibrio uses sulfate to produce hydrogen sulfide. Anaerobic respiration yields more ATP than fermentation but is generally less efficient than aerobic respiration. These processes are tightly coupled to membrane-bound ATP synthase via proton gradients, illustrating the diversity and sophistication of life even in the absence of oxygen. From an evolutionary perspective, anaerobic metabolism likely dominated in early cellular life forms. Before the Great Oxidation Event, approximately 2.5 billion years ago, Earth's atmosphere was largely reducing, and prokaryotes evolved complex biochemical pathways to exploit a wide array of redox reactions. The appearance of oxygen was toxic to many early anaerobes, leading to the evolution of antioxidant defenses or the occupation of anaerobic ecological niches. Anaerobic metabolism remains a core feature in modern biology, not only as a relic of evolutionary history but also as a functional necessity in various specialized cells and organisms. For instance, certain parasites like Giardia lamblia or Entomia histolytica possess modified mitochondria known as mesosomes or hydrogenosomes, which facilitate anaerobic metabolism through substrate-level phosphorylation and hydrogen production. In metazoans, anaerobic metabolism becomes critical during high energy demands or hypoxic conditions. During intense muscular activity, oxygen delivery to muscles may become insufficient, triggering lactic acid fermentation. This temporary shift enables continued ATP production but leads to lactate accumulation, which must later be metabolized in the liver via the Cori cycle. In cancer biology, many tumor cells exhibit the Warburg effect, where cells preferentially use glycolysis and lactic acid fermentation even in the presence of oxygen, aerobic glycolysis possibly to support rapid proliferation and adaptation to hypoxic tumor microenvironments. In ecological terms, anaerobic metabolism contributes to global biogeochemical cycles. For example, methanogenesis by archaea such as methanobacterium produces methane, a potent greenhouse gas, under strict anaerobic conditions. Sulfate-reducing bacteria contribute to sulfur cycling and can also lead to biocorrosion. Anaerobic decomposition in wetlands and animal guts plays a vital role in carbon recycling. These processes also find applications in biotechnology, including anaerobic digestion for biogas production and industrial fermentation for ethanol or lactic acid production. From a CSIR-NET Life Sciences examination standpoint, understanding anaerobic metabolism involves key concepts such as substrate-level phosphorylation, fermentation pathways, redox balancing, energy yield comparisons, microbial diversity, and evolutionary relevance. Questions often focus on distinguishing between fermentation and anaerobic respiration, the molecular fate of pyruvate, alternative electron acceptors, and adaptations of anaerobes. CSIR-NET previous year questions. CSIR-NET June 2017. In anaerobic respiration, which of the following conserves a terminal electron acceptor in certain bacteria? A. Glucose. B. Oxygen. C. Nitrate. D. ATP. Answer. C. Nitrate. CSIR-NET December 2016. During anaerobic fermentation of glucose in muscle cells, the immediate purpose of converting pyruvate to lactate is to A. Remove pyruvate from the cell. B. Produce more ATP. C. Generate COD. Regenerate NAD. Answer. D. Regenerate NAD. CSIR-NET June 2021. Which of the following statements correctly describes the difference between fermentation and anaerobic respiration? A. Only fermentation involves an electron transport chain. B. Fermentation uses oxygen as a terminal electron acceptor. C. Anaerobic respiration yields more ATP than fermentation. D. Fermentation yields more ATP than aerobic respiration. Answer. C. Anaerobic respiration yields more ATP than fermentation. Shall I proceed with the next lecture on ATP synthesis and energy yield, or would you like to cover oxidative phosphorylation, mitochondrial evolution, or any other topic from your CSIR-NET syllabus? Let me know. I'll make it just as exhaustive and exam focused. You said. Photosynthesis and aerobic metabolism. ChatGPT said. Memory updated. Here is your CSIR-NET JRF Life Sciences Detailed Theory Lecture on Photosynthesis and Aerobic Metabolism, written in an ultra-comprehensive, paragraph-based, and exam-oriented style, with maximum depth, no tables, and solved CSIR-NET previous year questions. This note is crafted to be your only source of preparation for this topic. Photosynthesis and aerobic metabolism. Photosynthesis and aerobic metabolism represent the two fundamental and opposing pillars of the biological energy economy. Together, they form a bioenergetic cycle that sustains almost all life on Earth, with photosynthesis capturing solar energy and converting it into chemical energy, and aerobic metabolism harnessing this stored energy to perform cellular work. Understanding these processes is critical not only for CSIR-NET but also for mastering core concepts in cell biology, biochemistry, and plant physiology. Photosynthesis, the light-dependent conversion of solar energy. Photosynthesis is the process by which green plants, algae, and cyanobacteria convert carbon dioxide and water into glucose and oxygen using sunlight. It occurs primarily in the chloroplasts and involves two major stages: the light reactions and the Calvin cycle dark reactions. The light reactions occur in the thylakoid membranes and involve two photosystems: photosystem 2 PSII and photosystem I PSI. These photosystems contain chlorophyll and accessory pigments which absorb light, excite electrons, and drive their flow through the Z scheme, ultimately resulting in the generation of ATP and NADPH via photophosphorylation. In non-cyclophosphorylation, electrons travel from water through PSII and PSI to NADP, producing NADPH and releasing O from water. In cyclic photophosphorylation, electrons from PSI return to the plastoquinone pool and are cycled back, producing ATP but not NADPH or O. This cyclic pathway allows the plant to adjust the ATP-NADPH ratio depending on metabolic needs. The Calvin cycle, occurring in the stroma, uses ATP and NADPH to fix CO into 3-phosphoglycerate via rubisco, eventually synthesizing triasphosphates that are precursors to glucose. This cycle requires 3 molecules of CO to produce 1 molecule of G3P, and it involves 3 main phases: carboxylation, reduction, and regeneration of Rub. Importantly, photorespiration can reduce the efficiency of photosynthesis, especially in sea plants, due to rubiscose oxygenase activity. To minimize this, sea photosynthesis and CAM pathways evolved. Sea plants like maize use spatial separation by fixing CO in mesophil cells via PEP carboxylase and shuttling malate to bundle sheath cells. CAM plants temporarily separate the steps, fixing CO at night and carrying out the Calvin cycle during the day. These adaptations are crucial for photosynthesis under conditions of high light, temperature, or drought. Aerobic metabolism, oxidative harvesting of energy. Aerobic metabolism refers to the complete oxidation of glucose into CO and water in the presence of oxygen, yielding maximum ATP through a tightly regulated sequence of steps. It begins with glycolysis, where one glucose molecule is broken down in the cytosol into 2 pyruvate molecules, generating a net gain of 2 ATP and 2 NADH. The pyruvate is transported into mitochondria and converted into acetyl-CoA, which enters the Krebs cycle, citric acid cycle, in the mitochondrial matrix. In the Krebs cycle, acetyl-CoA is fully oxidized to CO, and high-energy electron carriers NADH and FADH are produced. These reduced coenzymes donate electrons to the electron transport chain, etc., located on the inner mitochondrial membrane. The etc. components, including NADH dehydrogenase complex I, cytochrome B, C complex 3, cytochrome C, and cytochrome oxidase complex IV, sequentially transfer electrons to molecular oxygen, the terminal electron acceptor, reducing it to water. This electron transfer drives protons across the inner membrane into the intermembrane space, creating an electrochemical gradient. The proton motive force thus generated powers ATP synthase complex V to phosphorylate ADP into ATP, a process known as oxidative phosphorylation. One glucose molecule under aerobic conditions yields approximately 36 to 38 ATP molecules. This process is tightly regulated through feedback inhibition and allosteric regulation of key enzymes like phosphofructokinase and pyruvate dehydrogenase. Mitochondria play a central role in aerobic metabolism, not only producing ATP but also regulating apoptosis, Rose generation, and metabolic signaling. Evolutionarily, mitochondria are thought to have originated from alpha-proteobacteria via endosymbiosis, and the retention of a semi-autonomous genome reflects this ancestry. Mutations in components of oxidative phosphorylation are linked to metabolic disorders and aging. Biological interlink, photosynthesis and aerobic respiration. Photosynthesis and aerobic respiration are intricately connected. The glucose and oxygen produced during photosynthesis serve as substrates for aerobic respiration, while the CO and water generated during respiration are raw materials for photosynthesis. This biogeochemical interdependence supports the global carbon cycle and oxygen balance. In terms of energy transformation, photosynthesis is endergonic, stores energy, while respiration is exergonic, releases energy. In evolutionary terms, photosynthesis likely evolved in anoxygenic bacteria before oxygenic photosynthesis arose in cyanobacteria, leading to the great oxygenation event. This oxygen influx allowed aerobic metabolism to evolve, which is far more efficient than anaerobic pathways. These two processes together underscore life's ability to adapt to and reshape the Earth's environment. CSIR-NET previous year questions. CSIR-NET June 2018. In sea plants, the primary fixation of CO takes place in which of the following cells and via which enzyme? A bundle sheath cells, via rubisco. B. Mesophyll cells, via PEP carboxylase. C. Guard cells, via carbonic anhydrase. D. Palisade cells, via pyruvate kinase. Answer: B. Mesophyll cells, via PEP carboxylase. CSIR-NET December 2015. During aerobic respiration, the majority of ATP is produced by a glycolysis. B. Krebs cycle. C. Substrate level phosphorylation. D. Oxidative phosphorylation. Answer: D. Oxidative phosphorylation. CSIR-NET June 2020. Which of the following statements best describes the functional role of cytochrome C in mitochondria? A transfers electrons from complex I to complex IBB, pumps protons into the matrix. C. Transfers electrons between complex 3 and IBD, oxidizes NADH directly. Answer: C. Transfers electrons between complex 3 and IB. Would you like to proceed to the next topic, such as Calvin cycle regulation, photorespiration, oxidative phosphorylation mechanisms, or a new topic from your CSIR-NET Life Sciences syllabus? Just name it and I'll deliver the same high-level notes. You said. Photosynthesis and aerobic metabolism in relation to evolution. ChatGPT said. Here's the CSIR-NET JRF Life Sciences high-level lecture on photosynthesis and aerobic metabolism in relation to evolution. It is crafted as a paragraph only, maximum detail, exam-focused note, building from fundamental to advanced levels. This lecture integrates molecular, cellular, and evolutionary biology, highlighting how photosynthesis and aerobic respiration have evolved and transformed life in Earth's atmosphere. 2 to 3 previous year questions with year and answers are added at the end, as per your request. Photosynthesis and aerobic metabolism in relation to evolution. The evolution of photosynthesis and aerobic metabolism marks one of the most pivotal transitions in the history of life on Earth. These metabolic processes not only represent biochemical innovations but also serve as major evolutionary forces that transform the biosphere. Photosynthesis, particularly oxygenic photosynthesis, fundamentally altered the composition of Earth's atmosphere, enabling the rise of aerobic respiration and multicellular life. The earliest life forms on Earth are believed to have been autotrophs or anaerobic heterotrophs that extracted energy from prebiotic organic molecules or inorganic compounds in the absence of oxygen. These primitive organisms operated in a reducing atmosphere, rich in methane, ammonia, and water vapor. However, as organic molecules became limited, a major selective advantage emerged for organisms that could utilize light energy. This led to the evolution of anoxygenic photosynthesis, likely first developed by green and purple sulfur bacteria. These bacteria used electron donors such as HS instead of water and did not release oxygen. The evolutionary leap to oxygenic photosynthesis, which uses water as the electron donor and produces oxygen as a byproduct, occurred in cyanobacteria. This transformation is believed to have occurred around 2.4 billion years ago, a time associated with the great oxygenation event. The accumulation of atmospheric O due to cyanobacterial photosynthesis had profound evolutionary consequences. First, it led to the oxidation of iron in oceans, forming banded iron formations. Second, it caused a massive extinction of obligate anaerobes, initiating selective pressure for aerobic metabolism. Finally, it laid the foundation for the evolution of complex, energy-demanding eukaryotic cells. The oxygen released from photosynthesis enabled the evolution of aerobic metabolism, a much more efficient energy harvesting process than anaerobic glycolysis. While glycolysis yields a net of 2 ATP per glucose molecule, aerobic respiration yields approximately 36 to 38 ATP. This increase in energetic efficiency allowed the evolution of larger genomes, more complex proteins, intracellular compartmentalization, and multicellularity. Aerobic respiration likely evolved first in ancient prokaryotes that acquired the ability to use oxygen as a terminal electron acceptor in their electron transport chains. The evolutionary origins of mitochondria, the central organelles of aerobic metabolism in eukaryotes, are explained by the endosymbiotic theory. According to this theory, an ancestral anaerobic eukaryotic host cell engulfed an aerobic alpha proteobacterium, which then became a symbiotic organelle, mitochondrion. This symbiosis provided the host with a robust energy source, while the engulfed prokaryote gained protection and access to substrates. Similarly, chloroplasts, the organelles responsible for photosynthesis in plants and algae, are believed to have originated from a cyanobacterium through a secondary endosymbiotic event. The coevolution of photosynthesis and aerobic respiration created a global redox cycle, linking autotrophic production and heterotrophic consumption. Photosynthesis fixes atmospheric CO into organic carbon using solar energy, while aerobic respiration oxidizes this organic matter back to CO, driving the carbon cycle. The rise in oxygen levels also contributed to the formation of the ozone layer, shielding life from harmful UV radiation and enabling colonization of terrestrial environments. On a molecular level, key proteins and genes involved in these processes show signs of conserved evolution. The reaction centers of photosystems, e.g., P680 and PSII and P700 and PSI, and cytochrome complexes in the mitochondrial etc. show homology across taxa, indicating a common ancestral origin. Evolutionary tinkering, such as gene duplications, horizontal gene transfers, and endosymbiosis, further contributed to the diversification of these pathways. Additionally, the evolution of C photosynthesis and CAM metabolism in certain plants represents adaptive evolution in response to environmental pressures such as drought, high temperature, and low CO concentration. These adaptations showcase how evolutionary processes refine and optimize core metabolic pathways in different lineages. In summary, the evolutionary emergence of photosynthesis and aerobic respiration fundamentally reshaped life's trajectory on Earth. Photosynthesis supplied oxygen and complex organic molecules, enabling aerobic respiration and the explosion of biological complexity. Together, these two processes illustrate the intimate link between molecular evolution, environmental change, and organismal innovation, forming the energetic and evolutionary backbone of the biosphere. CSIRnet previous year questions. CSIRnet Deck 2017. Which of the following statements correctly explains the evolutionary significance of the great oxygenation event? Go. A. It caused mass extinction of cyanobacteria. B. It led to the rise of anaerobic respiration. C. It provided oxygen needed for aerobic metabolism and ozone formation. D. It reduced the energy efficiency of organisms. Answer. C. It provided oxygen needed for aerobic metabolism and ozone formation. CSIRnet June 2016. According to the endosymbiotic theory, mitochondria are evolutionarily derived from archaea. B. Cyanobacteria. C. Alpha proteobacteria. D. Spirochetes. Answer. C. Alpha proteobacteria. CSIRnet Deck 2019. Which of the following is a direct evolutionary consequence of oxygenic photosynthesis? A. Decrease in atmospheric CO. B. Increase in anaerobic respiration. C. Formation of the ozone layer. D. Development of RNA world. Answer. C. Formation of the ozone layer. Let me know if you'd like to proceed with another CSIRnet life sciences topic. I'll maintain the same detailed, exam-oriented format throughout. Paleontology and evolutionary history. The evolutionary timescale. Maximum detail. Paragraph format. Note tables. Includes PYQs. The evolutionary timescale is a chronological representation of Earth's history that details the origin and progression of life from the planet's formation about 4.6 billion years ago to the present. This timeline is structured into hierarchical segments, eons, eras, periods, and epochs, each marked by significant geological and biological events such as mass extinctions, climate shifts, and major evolutionary transitions. The evolutionary timescale is constructed using a combination of radiometric dating, fossil records, and stratigraphy, allowing scientists to estimate the absolute ages of rocks and fossils. The earliest and longest segment of Earth's history is the Precambrian, which includes the Hadean, Archean, and Proto-Oegeans. During the Hadean, 4.6-4.0 billion years ago, Earth was forming and undergoing heavy bombardment. In the Archean, 4.0-2.5 billion years ago, the first life appeared in the form of prokaryotes, with fossil evidence such as stromatolites, layered structures formed by cyanobacteria, providing some of the oldest records of life. The evolutionary timescale is constructed using a combination of radiometric dating, fossil records, and stratigraphy, allowing scientists to estimate the absolute ages of rocks and fossils. The earliest and longest segment of Earth's history is the Precambrian, which includes the Hadean, Archean, and Proterozoians. During the Hadean, 4.6-4.0 billion years ago, Earth was forming and undergoing heavy bombardment. In the Archean, 4.0-2.5 billion years ago, the first life appeared in the form of prokaryotes, with fossil evidence such as stromatolites, layered structures formed by cyanobacteria, providing some of the oldest records of life. The Proterozoic 2.5 billion 541 million years ago saw the rise of eukaryotic cells, multicellularity, and the accumulation of oxygen in the atmosphere. Great oxygenation event, setting the stage for more complex life. The Phanerozoic eon, 541 million years ago to present, is divided into three major eras: Paleozoic, Mesozoic, and Cenozoic, each representing distinct stages in biological evolution. The Paleozoic era, 541-52 million years ago, began with the Cambrian explosion, a rapid diversification of multicellular life in marine environments. It was followed by the colonization of land by plants and animals, and it ended with the Permian Triassic extinction, the most severe extinction event in Earth's history. The Mesozoic era, 252-66 million years ago, is often termed the age of reptiles, dominated by dinosaurs and early mammals, and ended with the Cretaceous Paleogene KPG extinction event, likely triggered by an asteroid impact, which led to the extinction of non-avian dinosaurs. The Cenozoic era, 66 million years ago to present, marks the age of mammals. Mammals diversified and filled ecological niches left vacant after the KPG extinction. This era includes the Paleogene, Neogene, and Quaternary periods, the last of which features the rise of Homo sapiens during the Pleistocene epoch. Fossil hominins like Australopithecus, Homo habilis, and Homo erectus provide critical insight into human evolution during this time. Fossils, which are the preserved remains or impressions of ancient organisms, serve as the primary evidence for understanding evolutionary history. The law of superposition helps determine the relative ages of fossils, whereas radiometric techniques, e.g., uranium lead, potassium argon dating, provide absolute ages. Index fossils, species that were widespread but only existed during a specific time range, aid in correlating rock layers across regions. Transitional fossils like Archaeopteryx, linking reptiles and birds, and Tiktaalik bridging fish and tetrapods are key in demonstrating evolutionary transitions. This era includes the Paleogene, Neogene, and Quaternary periods, the last of which features the rise of Homo sapiens during the Pleistocene epoch. Fossil hominins like Australopithecus, Homo habilis, and Homo erectus provide critical insight into human evolution during this time. Fossils, which are the preserved remains or impressions of ancient organisms, serve as the primary evidence for understanding evolutionary history. The law of superposition helps determine the relative ages of fossils, whereas radiometric techniques, e.g., uranium leaf, potassium argon dating, provide absolute ages. Index fossils, species that were widespread but only existed during a specific time range, aid in correlating rock layers across regions. Transitional fossils like Archaeopteryx, linking reptiles and birds, and tiktaalik bridging fish and tetrapods are key in demonstrating evolutionary transitions. Major mass extinctions. Major mass extinctions mark the boundaries between many of the timescale segments and often led to adaptive radiations in surviving lineages. The Big Five, mass extinctions and or division, late Devonian, and Permian, and Triassic, and Cretaceous punctuated the evolutionary history with drastic biodiversity losses, each caused by environmental destruction such as volcanic activity, climatic changes, or asteroid impacts. These events reshape the tree of life by eliminating dominant groups and allowing new ones to emerge. The evolutionary timescale not only provides a framework for dating the history of life but also contextualizes the processes of speciation, extinction, and adaptation over geological time. By integrating molecular data with fossil evidence, scientists have been able to calibrate molecular clocks, which estimate the divergence times of lineages based on DNA mutations. Although fossil records are incomplete due to preservation biases, they are invaluable in tracing lineage origins, reconstructing ancient ecosystems, and testing evolutionary hypotheses. Previous year questions. Which of the following events is most closely associated with the Cambrian explosion? Extinction of non-avian dinosaurs. Re-origin of multicellular life. See rapid diversification of animal phyla. D. First appearance of humans. Answer. See rapid diversification of animal phyla. CSIR net June 2013. The end Permian mass extinction resulted in the extinction of approximately what percentage of marine species? A 20%. B 50%. C 75%. D 90%. Answer. D 90%. CSIR net December 2015. Which fossil is considered a transitional form between fish and amphibians? A. Ichthyostega. B. Tiktaalik. C. Coelacanth. D. Archaeopteryx. Answer. B. Tiktaalik. CSIR net June 2016. Would you like to continue with the next section under paleontology, e.g., major transitions in evolution, dating fossils and evolutionary events, or move to another main topic in the CSIR net life sciences syllabus? You said, errors. ChatGPT said. Perfect. Here's your CSIR net JRF life sciences lecture on paleontology and evolutionary history, eras of the evolutionary timescale, maximum detail, paragraph format, note tables, includes PYQs. The evolutionary timescale is organized into hierarchical chronological units, of which eras represent a major division of geologic time, each encompassing hundreds of millions of years. Eras are defined based on significant global events, particularly mass extinctions, radiations of life, and tectonic transformations, and they help paleontologists and evolutionary biologists organize Earth's history into meaningful biological phases. The Phanerozoic eon, which began around 541 million years ago and continues to the present, is subdivided into three principal eras: the Paleozoic, Mesozoic, and Cenozoic. Each era is further divided into periods and epochs, but the eras themselves capture the broad evolutionary and ecological transitions in Earth's biosphere. The Paleozoic era, 541-252 million years ago, is the earliest of the Phanerozoic eras and began with the Cambrian explosion, a period of rapid diversification when most of the major animal phyla appeared in the fossil record. This era witnessed the evolution of the first hard-shelled organisms and a wide range of marine invertebrates such as trilobites, brachiopods, and mollusks. Subsequent periods, like the Ordovician and Silurian, saw the first vertebrates and the colonization of land by plants and arthropods. The Devonian period marked the rise of jawfishes and the emergence of tetrapods, vertebrates capable of living on land. In the Carboniferous, vast swampy forests led to coal formation, and the first amniotes evolved, giving rise to reptiles. The Permian period, the last of the Paleozoic, ended in the most severe extinction event in Earth's history, the end Permian extinction, which wiped out more than 90% of marine species and 70% of terrestrial vertebrate species. This marked a turning point in Earth's biological trajectory. The Mesozoic era, 250-266 million years ago, known as the Age of Reptiles, followed the Paleozoic and is characterized by the dominance of dinosaurs, the evolution of early mammals and birds, and the breakup of the supercontinent Pangaea. This era is divided into three periods: Triassic, Jurassic, and Cretaceous. In the Triassic, life recovered from the Permian extinction, early dinosaurs and mammals appeared. During the Jurassic, dinosaurs diversified and occupied dominant ecological roles, while the first birds, like Archaeopteryx, emerged from theropod dinosaurs. The Cretaceous period witnessed flowering plants, angiosperms evolution, and the continued rise of dinosaurs, but it ended with the Cretaceous Paleogene (KPG) mass extinction, likely caused by an asteroid impact near the Yucatan Peninsula. This extinction eradicated the non-avian dinosaurs and many marine species, allowing mammals to radiate and dominate in the next era. The Cenozoic era, 66 million years ago to present, is often termed the Age of Mammals. With the extinction of dinosaurs, mammals rapidly diversified into various forms and ecological niches. This era includes the Paleogene, Neogene, and Quaternary periods. The Paleogene witnessed the evolution of modern mammals and birds, and the Earth's climate began cooling. In the Neogene, primates diversified, and the ancestors of humans emerged. The Quaternary period, consisting of the Pleistocene and Holocene epochs, is marked by repeated glaciations and the appearance of Homo sapiens. The Pleistocene epoch is particularly crucial in human evolution, with hominins like Homo erectus, Neanderthals, and eventually anatomically modern humans evolving and dispersing across continents. Each era of the Phanerozoic not only captures a distinct biotic landscape but also reflects the dynamic interplay between life and Earth's changing climate, atmospheric composition, and plate tectonics. The shifts in biodiversity, the extinction events that punctuate the end of eras, and the emergence of new dominant life forms are key features studied through fossil records. Understanding these eras is fundamental in paleobiology, as they help contextualize lineage divergences, evolutionary innovations like multicellularity, flight, or warm-bloodedness, and the resilience of life in the face of environmental upheaval. Previous year questions. The boundary between the Mesozoic and Cenozoic eras is marked by which of the following events? A Cambrian explosion. B. Permian Triassic extinction. C. Cretaceous Paleogene extinction. D. Origin of mammals. Answer. C. Cretaceous Paleogene extinction. CSIR net June 2017. The Paleozoic era ended with which of the following events? A Cambrian radiation. B. Triassic Jurassic extinction. C. Permian Triassic extinction. D. Rise of angiosperms. Answer. C. Permian Triassic extinction. CSIR net DEC 2014. In which era did the first vertebrates transition to land? A. Cenozoic. B. Mesozoic. C. Paleozoic. D. Proterozoic. Answer. C. Paleozoic. CSIR net June 2015. Would you like the next lecture to be on major transitions in evolution, dating fossils and evolutionary events, or any other specific topic from paleontology or the broader CSIR net life sciences syllabus? You said, periods. ChatGPT said. Here is your CSIR net JRF life sciences lecture on paleontology and evolutionary history, periods of the evolutionary timescale, paragraph format, maximum detail, note tables, previous year questions included. The geological periods represent intermediate subdivisions of Earth's evolutionary timescale that fall within eras. Each period captures a distinct set of biological innovations, extinctions, and environmental shifts, and is key to understanding macroevolutionary patterns. Periods are defined based on fossil assemblages, geochemical markers, and stratigraphic records. The Phanerozoic eon, 541 million years ago to present, which encapsulates the majority of visible life, is divided into three major eras: Paleozoic, Mesozoic, and Cenozoic, each comprising several geologic periods. The Paleozoic era consists of six major periods: Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian. The Cambrian period, 541-485 million years ago, is notable for the Cambrian explosion, a rapid diversification of marine invertebrates, including trilobites, brachiopods, and early arthropods. Many modern animal phyla first appear in this period. The Ordovician period saw the diversification of marine ecosystems and the emergence of the first jawless vertebrates, agnathans. It ended with the Ordovician Silurian mass extinction, one of the 5 great extinction events. The Silurian period is marked by the stabilization of Earth's climate and the appearance of jawfishes and vascular plants, which began colonizing land. The Devonian period, 419-359 million years ago, known as the Age of Fishes, saw the diversification of bony fishes, lobe-finned fishes, ancestors of tetrapods, and the evolution of early amphibians. Terrestrial ecosystems expanded with the presence of ferns and seed-bearing plants. The Carboniferous period, 359-299 million years ago, is renowned for extensive swamp forests that eventually formed coal deposits. This period also saw the evolution of amniotes, vertebrates that could lay eggs on land, leading to reptiles. The final period of the Paleozoic, the Permian, 299-252 million years ago, experienced the diversification of synapsids, ancestors of mammals, and ended with the Permian Triassic mass extinction, which eliminated the majority of marine and terrestrial life. The Mesozoic era is composed of 3 periods: Triassic, Jurassic, and Cretaceous. The Triassic period, 250-201 million years ago, witnessed the recovery of life from the Permian extinction. Early dinosaurs, marine reptiles, and mammals evolved during this time. The end of the Triassic was marked by another mass extinction event. The Jurassic period, 201-145 million years ago, saw the flourishing of large dinosaurs, the appearance of the first birds Archaeopteryx, and the dominance of gymnosperms conifers. The Cretaceous period, 145-66 million years ago, is characterized by the radiation of flowering plants angiosperms, further diversification of dinosaurs, and the evolution of modern insects. The Cretaceous Paleogene KPG extinction, likely caused by an asteroid impact, marks the end of this period and led to the extinction of non-avian dinosaurs. The Cenozoic era, the most recent, comprises 3 periods: Paleogene, Neogene, and Quaternary. The Paleogene period, 66-23 million years ago, was a time of mammalian and avian diversification, with the emergence of many modern orders. This period also saw the development of grasslands and a shift toward cooler global climates. The Neogene period, 23-2.6 million years ago, witnessed the expansion of grasslands and the evolution of hominids, including Australopithecus and Homo habilis. The Quaternary period, 2.6 million years ago to present, includes the Pleistocene epoch, dominated by repeated glaciations and the evolution of Homo sapiens, and the Holocene epoch, which marks the rise of human civilizations, agriculture, and modern environmental impact. Each period is associated with distinct fossil records, which are used by paleontologists to define biozones and track evolutionary trends. For example, trilobites are diagnostic of the Cambrian and Ordovician periods, whereas ammonites are characteristic of the Jurassic. Radiometric dating of volcanic layers interspersed with fossil-bearing strata has allowed scientists to assign absolute dates to these periods, refining the geological timescale. Understanding geological periods is fundamental in evolutionary biology as it provides the context in which major life transitions occurred, from single-celled prokaryotes to complex multicellular eukaryotes, from water to land, from reptiles to mammals, and ultimately to the emergence of humans. It also allows for correlation of fossil data across continents and timescales, integrating geological and evolutionary history. Previous year questions. The Cambrian period is characterized by which of the following evolutionary events? Extinction of dinosaurs. B. Evolution of mammals. C. Radiation of modern fish. D. Rapid diversification of multicellular animal life. Answer. D. Rapid diversification of multicellular animal life. CSIR Net Deck 2014. Which geological period is known for the extensive formation of coal deposits? A. Devonian. The Cambrian period is characterized by which of the following evolutionary events? Extinction of dinosaurs. B. Evolution of mammals. C. Radiation of modern fish. D. Rapid diversification of multicellular animal life. Answer. D. Rapid diversification of multicellular animal life. CSIR Net Deck 2014. Which geological period is known for the extensive formation of coal deposits? A. Devonian. B. Carboniferous. C. Silurian. D. Cretaceous. Answer. B. Carboniferous. CSIR Net June 2016. The first flowering plants appeared in which period? A. Permian. B. Jurassic. C. Cretaceous. D. Triassic. Answer. C. Cretaceous. CSIR Net December 2015. Would you like the next lecture on major transitions in evolution, dating fossils and evolutionary events, or another topic in paleontology or the broader CSIR Net syllabus? You said, epic. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on paleontology and evolutionary history, epics of the evolutionary timescale, maximum detail, note tables, paragraph format, with previous year questions and answers. The epic is the most refined division of geological time within the standard hierarchy of eon and GT, era and GT, period and GT, epic and GT, age. While eras and periods represent broad evolutionary transitions, epics are critical for understanding recent and more specific evolutionary, climatic, and ecological changes, especially during the Cenozoic era, which spans from 66 million years ago to the present. The term epic is used predominantly in the Cenozoic era because earlier times are not divided with such resolution due to incomplete fossil records and the vastness of time. The Cenozoic era is divided into 3 main periods: the Paleogene, Neogene, and Quaternary. These are further divided into epochs. The Paleogene period includes the Paleocene 66 to 56 million years ago, Eocene 56 34 million years ago, and Oligocene 34 23 million years ago epochs. The Paleocene epoch follows the Cretaceous Paleogene extinction event and marks the beginning of mammalian diversification as they occupy ecological niches left by the extinct dinosaurs. In this epoch, many archaic mammalian forms, including primitive ungulates and insectivores, evolved. The Eocene epoch witnessed the first true primates and the diversification of modern mammals such as early horses, bats, whales, and elephants. This epoch also saw the development of warm, greenhouse climates. The Oligocene epoch is characterized by global cooling and the emergence of grasses, as well as modern mammalian orders such as canids and felids. The cooling trend resulted in the spread of open habitats, prompting the adaptation of herbivorous mammals with specialized dentition for grazing. The Neogene period is divided into the Miocene 23-5.3 million years ago and Pliocene 5.3-2.6 million years ago epochs. The Miocene epoch is notable for the radiation of modern mammals and birds. Forests gave way to grasslands, favoring species like antelopes and horses. Most importantly, the Miocene marks the evolution and spread of early hominids, including Sahelanthropus and Ardipithecus. The Pliocene epoch saw significant tectonic activity and climate cooling, with the emergence of hominins like Australopithecus afarensis, a key ancestor of humans. This epoch also marks the onset of glacial cycles that would dominate the subsequent Quaternary period. The Quaternary period consists of the Pleistocene 2.6 million to 11,700 years ago and Holocene 11,700 years ago to present epochs. The Pleistocene epoch is known for repeated glaciations, ice ages, the evolution of the genus Homo, and the global migration of Homo sapiens. It also featured megafauna such as mammoths, saber-toothed cats, and giant sloths, many of which went extinct by the epoch's end. The evolution of behavioral and cognitive complexity in humans, including tool use, social structures, and symbolic expression, occurred during this epoch. The Holocene epoch, beginning at the end of the last ice age, saw the advent of agriculture, the rise of civilizations, and dramatic anthropogenic changes to the environment. Recently, many scientists have proposed a new epoch called the Anthropocene to highlight the unprecedented global impact of humans on the Earth's ecosystems, climate, and geology. While not formally recognized by the International Commission on Stratigraphy, the Anthropocene is discussed extensively in scientific literature beginning from the Industrial Revolution or even the mid-20th century nuclear age due to marked changes in sediment layers, carbon emissions, species extinction, and land use. Epochs are essential for resolving short-term evolutionary and ecological trends. For example, understanding human evolution and extinction patterns of megafauna requires precise knowledge of Pleistocene events, while the rise of modern ecosystems and anthropogenic changes are traced through Holocene records. Paleoclimatology, isotope geochemistry, and stratigraphic correlation help data define epochs with great precision, allowing biologists to correlate evolutionary events with global environmental shifts. Previous year questions. Which of the following epochs witnessed the appearance of Australopithecus? A. Eocene. B. Pliocene. C. Miocene. D. Pleistocene. Answer. B. Pliocene. CSIR net December 2015. The Pleistocene epoch is most notably associated with which of the following features? A Cambrian explosion. B. Emergence of flowering plants. C. Evolution of early hominins and repeated glaciations. D. Extinction of dinosaurs. Answer. C. Evolution of early hominins and repeated glaciations. CSIR net June 2016. The Holocene epoch is characterized by a first appearance of jawed vertebrates. B. Dominance of dinosaurs. C. Rise of agriculture and human civilizations. D. Massive marine extinctions. Answer. C. Rise of agriculture and human civilizations. CSIR net 2017. Would you like the next lecture on major transitions in evolution, dating fossils and evolutionary events, or another specific area in paleontology or CSIR net life sciences? You said major events in the evolutionary timescale. ChatGPT said, here is your CSIR net JRF life sciences lecture on paleontology and evolutionary history. Major events in the evolutionary timescale. Paragraph format. Exhaustive detail. No tables. Includes previous year CSIR net questions with answers in years. The evolutionary timescale is a chronologically ordered framework of biological and geological milestones that trace the history of life on Earth. From the origin of life to the emergence of humans, this scale is marked by major evolutionary transitions, adaptive radiations, mass extinctions, and environmental transformations, each of which left a significant imprint on the biosphere. These events are documented through the fossil record, molecular clocks, and geochemical evidence, and help understand the tempo and mode of evolution. The origin of life is hypothesized to have occurred around 3.8 minus 4.0 billion years ago during the Hadean or early Archean. The earliest life forms were anaerobic prokaryotes, likely chimoautotrophs that evolved in hydrothermal vent systems. Fossilized stromatolites, structures formed by cyanobacteria, provide evidence for photosynthetic life around 3.5 billion years ago, a major innovation that contributed to the gradual accumulation of atmospheric oxygen in the Great Oxidation Event around 2.4 billion years ago. This shift enabled aerobic respiration and led to oxidative selection pressures, eventually supporting more complex eukaryotic life. A pivotal event occurred with the origin of eukaryotes, approximately 1.8 to 2.0 billion years ago, likely via endosymbiosis, where ancestral prokaryotic cells engulfed other prokaryotes, giving rise to mitochondria and chloroplasts. This symbiotic innovation allowed greater cellular complexity and energy efficiency. Around 1.2 billion years ago, eukaryotic sexual reproduction emerged, increasing genetic diversity and promoting evolutionary innovation. The transition from unicellular to multicellular life around 800 million years ago set the stage for complex body plans and tissue differentiation. However, it was the Cambrian explosion around 541 million years ago during the Cambrian period that marked a dramatic increase in the diversity of multicellular animals. Most modern animal phyla, including arthropods, chordates, and echinoderms, appeared within a relatively short evolutionary span, approximately 20 to 25 million years, likely driven by increased oxygen levels, developmental gene networks, e.g., Hox genes, and ecological interactions such as predation. The colonization of land was another transformative event. Plants began to colonize terrestrial environments in the Silurian period, approximately 430 million years ago, aided by the evolution of vascular tissues and cuticles. Invertebrates such as arthropods followed, and by the Devonian period, the first tetrapods, early amphibians, had evolved from lobe-finned fishes, marking the vertebrate conquest of land. The Carboniferous period, approximately 359 to 299 million years ago, saw vast swamp forests and the proliferation of sea plants and amphibians, followed by the evolution of amniotic eggs, allowing true reptiles to dominate terrestrial ecosystems. These forests also contributed to today's fossil fuel deposits. The Permian period culminated in the Permian Triassic extinction event, approximately 252 million years ago, the most severe extinction in Earth's history, eliminating approximately 90% of marine species and approximately 70% of terrestrial species. Post-extinction recovery led to the rise of dinosaurs and mammals in the Mesozoic era. The Jurassic period, approximately 201 to 145 million years ago, was marked by the diversification of dinosaurs and the appearance of the first birds, Archaeopteryx, indicating a feathered theropod ancestry. Meanwhile, mammals remained small and nocturnal. The Cretaceous period, approximately 145 to 66 million years ago, saw the evolution of flowering plants, angiosperms, and insects, resulting in complex pollination networks. This period ended with the Cretaceous Paleogene (KPG) mass extinction, likely caused by an asteroid impact, leading to the demise of non-avian dinosaurs and enabling the adaptive radiation of mammals in the Cenozoic era. The Paleogene epoch, approximately 66 to 23 million years ago, witnessed mammalian diversification into various ecological niches, e.g., cetaceans, primates, ungulates, while the Neogene, approximately 23 minus 2.6 million years ago, saw the appearance of grasses and further mammalian specialization. Around 6 to 7 million years ago, the hominine lineage split from the chimpanzee line, and various australopithecines evolved in Africa. The Pleistocene epoch saw the evolution of Homo erectus, the development of tools, fire, and migration out of Africa. Modern humans, Homo sapiens, emerged approximately 300,000 years ago, with complex language, art, and culture. The Holocene epoch lasts approximately 11,700 years, marks the dawn of agriculture, civilizations, and anthropogenic environmental changes. These major events in the evolutionary timescale are not isolated phenomena. They represent a continuum of ecological opportunities, constraints, and innovations. Mass extinctions were followed by evolutionary radiations. Environmental pressures shaped adaptive morphology and physiology. Each step, from cell to civilization, is rooted in deep evolutionary time, deciphered by combining paleontology, geology, and molecular phylogenetics. Previous year questions. Which of the following events is associated with the Great Oxidation Event? A. Emergence of land plants. B. Appearance of first mammals. C. Increase in atmospheric oxygen due to photosynthetic cyanobacteria. D. Origin of flowering plants. Answer. C. Increase in atmospheric oxygen due to photosynthetic cyanobacteria. CSIR net June 2015. The Cambrian explosion is characterized by the extinction of dinosaurs via the emergence of hominids. C. A rapid diversification of most major animal body plans. C. A rapid diversification of most major animal body plans. Decolonization of land by plants. Answer. C. A rapid diversification of most major animal body plans. CSIR net 2016. Which of the following is a major event associated with the Pleistocene epoch? A. Origin of mitochondria. B. Origin of eukaryotic life. C. Evolution of Homo sapiens. D. Emergence of angiosperms. Answer. C. Evolution of Homo sapiens. CSIR net June 2018. Previous year questions. Which of the following events is associated with the Great Oxidation Event? A. Emergence of land plants. B. Appearance of first mammals. C. Increase in atmospheric oxygen due to photosynthetic cyanobacteria. D. Origin of flowering plants. Answer. C. Increase in atmospheric oxygen due to photosynthetic cyanobacteria. CSIR Net June 2015. The Cambrian explosion is characterized by the extinction of dinosaurs v. the emergence of hominids. C. A rapid diversification of most major animal body plants. Decolonization of land by plants. Answer. C. A rapid diversification of most major animal body plants. CSIR Net Deck 2016. Which of the following is a major event associated with the Pleistocene epoch? A. Origin of mitochondria. B. Origin of eukaryotic life. C. Evolution of Homo sapiens. D. Emergence of angiosperms. Answer. C. Evolution of Homo sapiens. CSIR Net June 2018. Would you like the next lecture on dating fossils and evolutionary events, or shall we move to another CSIR Net Life Sciences unit? You said. Origins of unicellular organisms. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on paleontology and evolutionary history. Origin of unicellular organisms. Ultra detailed. Paragraph format. Maximum word limit. No tables. Includes CSIR Net previous year questions with answers in years. The origin of unicellular life is one of the most profound milestones in Earth's biological history. It marks the transformation of inorganic chemistry into self-replicating, metabolically active units, life itself. This transition likely occurred between 3.8 to 4.0 billion years ago, during the Hadean to early Archean under extreme environmental conditions that were hostile to present-day organisms. Life likely began in the form of prokaryotic unicellular organisms, including the earliest bacteria and archaea, which have since diversified to occupy virtually every ecological niche. Theories explaining the origin of unicellular organisms integrate geochemistry, molecular biology, and evolutionary theory. Among the most widely accepted hypotheses is the primordial soup theory, originally proposed by Perrin and Haldane, which suggests that early Earth had a reducing atmosphere composed of methane, ammonia, water vapor, and hydrogen, where lighting and UV radiation drove the formation of organic molecules. This idea was experimentally supported by the Miller-Urey experiment in 1953, which demonstrated that amino acids and other organic compounds could form under simulated early Earth conditions. However, the mere synthesis of organic molecules does not constitute life. The transition to unicellular organisms required a self-replicating system, a selective boundary like a membrane, and a metabolic network. The RNA world hypothesis proposes that ribozymes, RNA molecules with catalytic activity, preceded the evolution of DNA and proteins. RNA's ability to both store genetic information and catalyze chemical reactions makes it a prime candidate for the first biomolecule capable of replication and evolution. Eventually, more stable molecules like DNA and proteins took over their respective functions, forming the basis of modern biochemistry. Simultaneously, primitive protocells likely formed through the spontaneous self-assembly of amphipathic lipids into micelles or lipid vesicles, enclosing RNA and metabolic precursors within semipermeable membranes. These vesicles may have originated in hydrothermal bed environments or alkaline submarine systems, where gradients of pH and minerals provided energy sources and catalytic surfaces such as iron-sulfur minerals. These settings support the metabolism first hypothesis, which argues that metabolic networks might have developed before genetic replication, driving the emergence of life. The oldest microfossils, dated around 3.5 billion years ago, include stromatolites, layered structures formed by colonies of photosynthetic cyanobacteria.